adipocyte, then you store it. So if we're in an adipocyte, so one way to get the fats into your cells for burning them right away is to get them through the diet. Another way to get fats to access them for burning is to recover them from storage. Right. So this is kind of like breaking down glycogen, but now we're like recovering fat. So the fat has not polymerized into some like maxi fat, highly polymerized branched molecule like glycogen. It's simply in a certain sort of condensed as a globule, a lipid droplet in the fats, and it tends to be in the fat cells. Right? So we have a hormone cascade to actually mobilize the fatty acids here, right, to get them here into the bloodstream, and then possibly also packaged into LDLs and then delivered. So we have fat cells in the body that respond to these hormones. Low blood sugar hormone, glucagon, right? Therefore, we need to mobilize. We need energy. Cascade, protein kinase A, adenyl cyclase, protein kinase A, phosphorylating a lipase. Right? So a lipase here, hormone-sensitive lipase, HSL, is used to break up the fats and generate the free fatty acids here. Right? So there's different pathways here. There are these molecules called perilipin that get phosphorylated by pKa as well. And perilipin is a sort of amphipathic molecule that helps to break up the lipids so that lipases can access them better. And then you get mobilized fatty acids in the blood. You can make LDLs out of them, but another way to transport them is to bind them to a blood protein called serum albumin to transport them that way. Okay. So just so we have a sense that yes, fat can get stored, and if we want to get energy, we have hormone-based ways to break it down and then deliver the lipids to cells where they're needed. Okay. So now we're in the cells, we've got the fatty acids in there, and we want to break them down and get energy out of them. Okay. So how do we do that? So here's the fatty acid in red. Okay. So very easy to draw. There's just some R group with a carboxylate on the end. And so the first thing that happens is that the fatty acid is activated, and so there's a kind of like in glycolysis where the glucose is, you invest ATP. The equivalent here would be investing ATP to activate the fatty acid, and it's activated into a fatty acyl-CoA molecule. Okay. So how this works is the carboxylate oxygen of the fatty acid attacks the alpha phosphorus of ATP, releasing pyrophosphate, which is then cleaved by pyrophosphatase to generate 2 molecules of inorganic phosphate, which is favorable. And so this attack with the release of pyrophosphate generates a fatty acyladenylate, which is bound to the enzyme, the fatty acyl-CoA synthetase. So it's the same enzyme here, just 2 steps. Right? So it binds 3 substrates, ATP, fatty acid, and then after that adenylate is formed, coenzyme A binds, and the sulfur attacks this carbon, AMP is released, so this is also activated. AMP is a good leading group too. You end up with fatty acyl-CoA. So you've invested how many ATPs?
2.
2 equivalents of ATP. Right? That discussion was not in vain. So AMP, right, you would need 2 to break to go back up. And so we will see in a few slides when we do the arithmetic that this is costing -2. But what we also get out of it is a thioester, which is a nice high energy bond. So that's good. Okay. So this is generating the fatty acyl-CoA, and that's the key thing we have to have. So the fatty acyl-CoA is generated in the cytosol. So that last reaction was a cytosolic reaction. Actually, let me give you this analogy. It's too interesting to me to not to sort of pass up. So if you are a coding enzyme in the cell that needs to generate amino-acylated tRNA to express a genetic code, then you will bind ATP. Instead of fatty acid, you'll bind amino acid. Instead of a fatty acyladenylate, you'll generate an amino acyladenylate. And instead of being attacked by CoA, you'll be attacked by a transfer RNA molecule. An OH group at the very 3' end of a tRNA molecule, and you'll generate amino acyl tRNA instead of fatty acyl CoA. But the steps are exactly the same in terms of the sequence of events, in terms of the chemistry. It's not sulfur, it's oxygen on tRNA, but that two-step process of amino acylation is exactly analogous to this. Okay. So you'll get that next term, for sure. All right. The fatty acyl CoA in the cytosol, but most of the burning of the fat is going to happen, of the fatty acid is going to happen in the mitochondria for reasons that are should be apparent, because we're going to be generating a lot of reducing equivalents that are going to be useful to generate ATP by oxidative phosphorylation inside the mitochondria or on the inner mitochondrial membrane. So we want to take this energy-laden group here, this fatty acyl CoA with this nice long hydrophobic side chain, and get it into the mitochondrial matrix here. And so the way to do that is that there's a specialized transport system. And this is how fats are regulated between being synthesized, which they get synthesized in the cytosol, and being broken down in the matrix. Right? So normally this is where fatty acids get synthesized, right? So, but it's also where we bring them in from the outside. So as we bring them in, we want to rapidly esterify them to CoA, trans-esterify them to a molecule called carnitine, which is just a small molecule, 4 or 5 carbons and some other stuff. You can look at the structure in the book, it's not that important. But CoA will leave and carnitine will be acylated on, and then there's transporters to get this through the membrane and then to get this into the intermembrane space. Sorry, you don't need a transporter here because it's relatively porous, but you need an enzyme to put carnitine on it. Carnitine will go through the intermembrane space, and then there's a specific transporter here to get the carnitine acylated fatty acid here into the matrix, and then coenzyme A in the matrix regenerates fatty acyl CoA. So fatty acyl CoA is the compound that goes into the burning of fatty acids, but we have to trans-esterify it, move it through the membrane, and move it through, take it back to CoA inside the mitochondria. Okay. Questions?
Is that because the CoA molecule is just too large to pass through the membrane?
It's probably not because it's too large. It's just because we want to provide a way in which we regulate the process of the burning inside the mitochondria with synthesis, with respect to synthesis in the cytosol. And on Friday we'll talk about that, and then that will show you a role for carnitine that basically has to do with regulation. Yeah. Okay. So, all right. So we're ready to talk about beta oxidation. And the input to beta oxidation is a fatty acyl CoA. This is just giving you the overall cycle, right? So we're going to have, so for example here, we've got 16 carbons, right, in a fatty acid side chain. It's not showing you the CoA here, right? But, so stage 1 will be this process of beta oxidation that we're about to talk about, okay? And what that's going to do is it's going to take this C16 fatty acid, and it's going to generate 8 molecules of acetyl-CoA. So we've got, this is 2 carbons, this is 16, so we need 7 steps, right? Not 8. We need 7, because when we get down to 4, we split it, we get 2 out of that, so we need 7 passes through beta oxidation to generate 8 acetyl-CoAs from 1 C16 fatty acid. And the example we're going to use is a nice, long, saturated, straight C16 fatty acid. That'll be the first example, and then there'll be some convolutions around what happens if there's double bonds and then things like that. So now we've generated 8 acetyl-CoAs, which of course can shoot right into the TCA cycle. So most of the energy you get out of the fats is actually coming from the TCA cycle. So the TCA cycle is both a sugar accepting cycle and a fat accepting cycle. Right? I mean, it accepts acetyl-CoA in either case, but acetyl-CoA comes from fats or it comes from glucose. Right? So that's the beauty of the centrality of the TCA cycle, is that you can feed acetyl-CoA in from both of your major both of your major energy molecules. And then stage 3 is making ATP by oxidative phosphorylation, which of course becomes the topic of the class after we're through with the next exam. Okay, so the first step, here's the C16, palmitoyl-CoA. Palmitate is just a common name for C16. You don't have to remember that. So the first step is the oxidation of the fatty acid, alpha beta to the thioester linkage here. Right? So that's important. So you can draw an R, and draw a few carbons out, right? And then CoA is esterified just through here, and then that's where the bond goes in. Acyl-CoA dehydrogenase, oxidation, reduction of what? Turns out it's FAD, FADH2. Acyl-CoA dehydrogenase generates FADH2, and the electrons are funneled into the electron transport chain at Q is ubiquinone, which is the cofactor in electron transport that we'll talk about later. But for now, enough to know that the reducing equivalents are going to go into the electron transport chain. So notice that this reaction here is highly analogous, to put it mildly, to this succinate dehydrogenase reaction here, which is the TCA cycle, right? TCA cycle, beta oxidation. First step, right? This is step 6 of the TCA cycle.